Thank you for the kind introduction. Before I start telling you about the tool that we are developing at Mount Sinai, I want to give you a little bit of information about our research lab. We are a cancer genomics lab where we aim to identify and target drivers of cancer genesis, progression, and therapy. And this is a word cloud of some of the work that we've done from the abstracts. So you can see it's mostly cancer and networks and so forth. And while doing these research, we have been developing software and web interfaces for data exploration, and these are some of them. And in our work, we collaborate with clinical and experimental and computational groups. And some of the NIH-sponsored consortiums we collaborate with is Human Immunology Project Consortium, HIPC, CPTech, which is a clinical proteomics tumor analysis consortium. And the work I'm going to present today is with the CIMAC-CIDC, which is the immuno-oncology consortium sponsored by the NCI. And in our work, what we want to do is to be able to build bridges between data viz and cancer. And we are at the interface of different areas, for example, clinical oncology research and basic research on one end, and on the other hand, computational oncology, web development, and visualization design. And we try to know a little bit of each one of them and maybe master of none. But the PRIMAVO, the tool that we are developing, is sponsored by President Biden's Cancer Moonshot Initiative. With the Cancer Immune Monitoring and Analysis Centers, CMAX, and the Cancer Immunological Data Commons, CIDC. And the objective of these Cancer Moonshot efforts is to be able to figure out who's going to benefit the most from cancer immunotherapy clinical trials. And in order to be able to do that, so we can, instead of looking at a population and giving all of them immunotherapies, we can stratify the patients, and we can also predict who is going to respond the best. And in order to do that, these centers are collecting disparate immune monitoring omics data sets, and these are plenty: protein expression, gene expression, cell abundance, antibody levels, genetic mutations, microbiome analysis, immunohistochemistry, and most recently even spatial sequencing. And not only are we swimming in data from these different analytes, we also have the problem that, or the opportunity that these measurements are done in multiple time points at baseline immunocompetence, dynamic changes in early treatment, and immune correlates after treatment. So there's a lot of data to be able to explore and interact with. And we developed PRIMAVO as a web-based tool to visually analyze and interactively explore immune monitoring assay results from these cancer moonshot immunotherapy clinical trials. And in the back end, we have data coming from CMAC and CIDC and different data types based on the different analytes. And in the front end, we have three different sections. One of them is the study selection, where we select between different clinical trials. And once we select the clinical trial, then we go to the data selection, where we can filter the patients we're interested in using different criteria. And we can also then go to the data exploration page that I'm going to show you in a little bit, where we can cluster, normalize, zoom, etc., do a lot of different things using different visualization options. And we do have tutorials, user feedback, and FAQ. And however, the tool is currently not available publicly because of NCI's extremely strict data access policies, and I'm going to show you a little more details of PRIMAVO based on synthetic data. I cannot even show you one clinical trial data, but they're trying to resolve this. And PRIMAVO is going to be hosted in the NCI server, and whoever has access to these data sets will be able to have access. However, with that said, we're also going to have a freely available version of PRIMAVO so that other individuals who don't have these extreme data access restrictions are able to access them. And so first we have a study selection page, which is just like we have like a very simple interface where users can choose between different clinical trials, and also every trial, what kind of analytes are available. And also we can have more information going to the clinical trials gov website. And from the data, once we select the clinical trial, then we go to filter for specific criteria. And all of these charts that you see in the data selection page are completely interactive, and they update in real time. So these could be therapies, assay, cancer types, responses, ethnicity, mutated genes, stage, treatment, drug, sex, number of advanced events, age, tumor mutation burden, survival. All of these updated in real time. I'm going to show you in demo. And once we filter the patient based on the criteria that we are, And so in this page where, for example, where we have the patient filtering, so you can see now in the video how many patients we have, and then on the left we have the arms. So for example, in which arm of the study of the clinical trial, what is the drug dosage, how many patients, what is the drug combination, was it escalation, and so forth. And then we can select between different assays or cancer types, and for example, the different responses. So if we select, for example, partial response, then all the data is updated for patients that had partial response. And we can go even further, filter more the data that we have based on the whatever specific criteria that we are interested in. For example, we can say, oh, I'm only interested in partial response, but white patients, and then we can filter it and then get the information. And whatever data we select, then when we go to the next page, it's going to update accordingly. I'm going to fast forwarding in the interest of time. And for example, if we are only interested in patients that are older than 42, we can do that too. And then we can also remove all the filters if we want to. And once we select the patients, we can also look at, for example, survival information of these patients, and then compare survival based on sex, compare survival based on treatment type, and we can even combine different treatments and have a completely so we basically have a completely interactive survival plot where we can explore the data more. And once we do our data selection and filtering, what we do is we go to the data exploration page where we have interactive heat maps for example, for cytoph, O-link, serology, and gene expression assays. And on the left heat map, the x-axis are the markers that we're measuring, and then the y-axis is the patients and with their de-identified IDs. And on the right heat map, we have a summary, which is comparisons across all study arms and treatments, so that we can look at the differential expression of genes or protein abundance, for example, and see which experiments are which treatments are the most different among which treatments are different. And when we, for example, from the site of patient marker data analysis, what we can do is we can select a specific marker, and if we click on it, then a box opens up and we can see how this marker has changed over time across different treatments, and we can all do all of these in real time also. And if we have the video, a demo of it, for example, and in the data exploration page, all the different analytes are in top, cytoph, serology, O-link, RNA-seq, IHC, and whole exome sequencing. And then here, this page is the cytoph data that we have, and in the cytoph, the heat map, we can, we can interact with it however we want to. For example, on the x-axis, we have the markers. On the y-axis, we have the patients. And we can cluster this heat map in real time based on different clustering criteria. And the heat map updates in real time. And then once we cluster it, we can also look at it in further detail of, for example, the details of the markers that we have. And also we can order, or for example, we can order them, for example, with respect to treatment, and then everything changes in real time. Again, if we click on sex, then it reorganizes the heat map. So there are a lot of different ways of looking at this heat map, which is completely interactive in order to be able to explore the data that we're interested in. In the interest of time, I'm just going to fast forward this. And okay, yeah, and other things that we can do is, for example, we can also normalize, like we can have the raw data, and now we just z-normalized it, so we can also normalize it based on whatever the user wants. So all the heat maps are completely interactive, and then once it's normalized, we can explore how the different entities change within the heat map. And we can pick a marker, and once we pick the marker, then we can have the in real time whatever marker we pick, how it changes in real time in terms of different measurements over time of the patients. And then these boxes are also completely interactive. We can modify them based on the different marker, different treatment. Maybe we want to see how we want to reorganize the patients and see their time course in a different way. We can do all of these interactively in real time. So this is just the data, so the site of raw data. And then we can also, and then if we go to the summary data analysis, where we have the treatment comparisons. For example, if we look at the partial response versus progressive disease, and then we can have like a volcano plot opening up, providing corresponding values of cell abstract markers in these groups. And here we have the volcano plots. These are not changed between the partial response and progressive disease patients. However, these are bad markers that we don't want to see, and these are the good markers. So if we see these markers, it's most likely that the patient is going to do well. And this again is summary heat map is also completely interactive that we can do. And it also has properties where the users can cluster them based on whatever their interests are, where they like in terms of rows or columns. And a few things that this also has is that we can also change the p-values. Maybe we want to have the threshold and not that strict or very strict. So we can, based on the p-value threshold, the values are going to change in real time, and we're going to see which changes are the most significant. And we can also organize with respect to response and time points and so on and so forth. And another thing we can do is if we select one of these, a little bit more detail, again, like now we have the volcano plot that I just mentioned to you in the previous slide, and now we can interactively explore this in more detail. And we can look at the very strict p-values, or we can also make it more adjusted p-value, have more of a flexibility, and see so different levels of interacting with the data all in real time. And let's go to the next one. Okay, and for whole exome sequencing data, we have standard oncoprints that are completely interactive, and the oncoprints display genomic mutations across study samples. And here you see the, for example, different properties, race, gender, other metadata that we have, and the top gene that was mutated in most tumors, P53, P10, BRCA1, and the percentages and the mutation types, all of these in oncoprint. Again, these are all interactive. And another property that we have within PRIMAVO is interactive charts for selecting regions of interest and immunohistochemistry data. And for example, here, if we have a region of interest from an IHC slide, what we have is then we can zoom in and pick a tile, and from these tiles, then we can look at different markers in more detail, and all of these are again interactive. And in the interest of time, I'm just going to show you very, very small interactivity video. And here, for example, we chose 2 markers, and with these 2 markers, we can look at it in more detail, or we can open different tiles and so forth. And so with that, PRIMAVO is a web-based tool to visually analyze and interactively explore immune monitoring assay results from cancer immunotherapy clinical trials. Results can be utilized for observational studies or inform subsequent preclinical studies and clinical trials. Users can filter, query, visualize, and share myriad high-dimensional assay data of study participants at multiple time points. It's fully customizable and flexible visualizations at both participant level and cohort level visual explorations to accelerate research in identifying biomarkers that can be linked to clinical outcomes. And in all throughout the design of PRIMAVO, we have followed user-centered design principles, which means that by continuously soliciting feedback from our end users at every step of its development, which was a massive amount of work. And with that, Osho Rawal is the main web developer and our main collaborator who has painstakingly answered all our questions related with data is Sasha Ginyatic and Edgar Gonzalez-Coslova. And I thank again President Biden and his Cancer Moonshot effort. Thank you.
Thank you very much for the talk. The floor is open for questions if there are any. And I would like to remind our online participants that feel free to ask your questions in the Q&A part in the chat. Yeah, they can be read here and answered.
Thank you very much. It's a very impressive amount of work that's gone into that. I just had a very basic question. Possibly you answered it and I missed it. What did you write this in? How did you make these plots?
Oh yeah, JavaScript, and we use different libraries. I think I have the slide. If we go back, so we have for the backend we have Python, Django, and from the front end we have. I mean, we used a lot of React, and yeah. But mostly it's all JavaScript.
Right, thank you.
Thank you.
Any more questions? Oh, it was not the case. I have one on my own. You have mentioned that you will be making the tool available online. Does it mean that I will be able to upload my own data and to test it on it?
Yeah, absolutely. That's our plan. We want to make it available. And we've talked to other investigators in the consortium who do not have NCI's extreme data access restrictions. And they are willing to share data with us that we can provide. And that's going to be our beta tester to how easy it is going to be to customize, like for the user themselves to upload data, because there are tables and tables of data. So we want to figure out how best to do that without disorienting the user. And of course, they're gonna need some help from us, but the objective is eventually to make this fully automated, and we'll see how far we can go with that.